welcome back to the Emerald. I thought she was about to open it up. I was gonna let you go.
Oh, I wanted to. I'll save that for the next one.
Okay, you can open the next one. Back at the Emerald Conference here. I call it Emerald Scientific, but Emerald Scientific is the company that originally started it, and that was the Emerald Conference. And then it got sold to MG BizCon, and then a company called Emerald has nothing to do with Emerald Scientific or the Emerald Conference bought it. So it's a little confusing, but it's the Emerald Conference, and it's one of the top conferences of getting together some of the intellectual thought leaders in the the scientific space, the development of cannabis in general. And we are pleased Dimitri Downing with
Destiny Blanco,
who's co-hosting Meet on Shackle today. We are pleased to be joined by another one of these wonderful individuals that are the attendees, speakers, and vendors here. Anna Schwabe.
That's me.
That's me. Yeah. I'm glad we got that right.
Yep, that's me.
Wait, that's not Anna Schwabe? All right. So Anna, Dr. Anna Schwabe, excuse me.
And we were just talking about this too. You can recognize her by her lovely pink hair.
Yes, definitely, it's very pink.
It is
I love the vibe.
pink. It's very bright.
No, I like it. I should go with pink hair. What do you think? Do you think I should? We
You
should
should
do
do some
that.
like highlights, like some cheetah print highlights.
No.
Maybe just a stripe, a block,
Oh, that would be cool.
Yeah, but
money piece.
Grow out like a little bit of long hair.
did Yoda have hair? Wait.
Just like 4 hairs.
Yeah, we have Yoda stowed away here somewhere.
Where is our Yoda?
Sorry about that. So we like to get into a little bit about your past, who you are, where you came from, where you got into cannabis, how you discovered the plant, the community, and what projects and stuff you're working on, etc. So let's start. We'll just go through your journey from the beginning. Where were you born?
I was born in Western Australia.
Oh, you're Australian.
I am Australian.
See, I wouldn't have known that if I didn't ask that.
No, it's super secret.
Oh, all
Nobody
right.
knows.
I could do an Australian accent, I think.
I was like, usually
I can, but I usually need a couple of drinks first, and then I
Oh,
can
yeah.
just slip right into it.
lived in America too long now.
Yeah.
Yes.
Since '87, we moved to Colorado.
1987, we arrived in Colorado.
Mhm.
We're getting closer to the cannabis stuff. Okay, so
So much closer.
what? I know. Well, we jumped.
Colorado too.
Look, you see how we jumped from birth to Colorado in '87. How old were you when you arrived in '87?
13, almost 13.
Okay, so maybe you started your college. Okay, we'll see what happened when you were 13. You arrived in Colorado. Then what happened next?
Well, I had technically finished and was in high school, like my first year of high school when we moved.
Yes.
But I was only 12 when we moved
Right.
here, so I
Mhm.
wasn't old enough to go into high school. So I had to kind of go back to 8th grade.
Okay.
And do a whole nother year of 8th grade, which was probably a good thing because the culture shock was real.
So you had to
Yeah.
wait another year to start your cannabis journey because no self-respecting high school kid in Colorado doesn't know about cannabis.
Yeah, it didn't start till high school.
That's like a 14, 15,
Yeah,
15.
Okay, so moving on from high school, you went to college. Then what?
I went to CSU for 5 years, and I was majoring in anatomy and physiology.
Anatomy and physiology.
And did not graduate, but was 12 credits short. I don't know. I just took a hiatus, had a baby, went
Nice.
back to school, and I transferred all my credits to UNC, the University of Northern Colorado. I have all 3 of my degrees from the University of Northern Colorado.
Excellent.
Awesome.
Yeah.
And when did that all finish up?
So let's see. I got my bachelor's degree in 2010 is when I graduated, and then 2012 I graduated with my master's. And I worked at the Denver Botanic Gardens for a couple of years, which is really where my interest in cannabis started because I had a volunteer in my lab that was in the industry, and we would have conversations.
And what year? About what year was that?
This was 2012 to 2015.
Okay, we are getting right hot
You're
into the
getting so close.
middle of cannabis activity now.
Mhm.
Colorado, 2012, 2013,
That's when
post-medicinal
That's when
legalization,
they legalized for adult use.
2013. Excellent. Yeah. So you're at Denver Botanical Gardens.
Yep. And so I had a volunteer, and we would have
Yes.
all these conversations, and the topic of differences in strains from dispensary to dispensary,
Right?
where there shouldn't be any difference. You know, if you're buying
Mhm.
Blue Dream, it should be the same everywhere. Just like if you buy a Snickers bar, it should be the same everywhere. You shouldn't,
Yeah.
you know. So I got the brilliant idea for this PhD project. And
Pulling out some props.
Okay.
Okay. Keep going.
I got this brilliant idea because my background is in plant population genetics.
Okay.
I could very easily answer that question.
But you weren't studying cannabis yet.
Not
But you started in agriculture plants and.
I started in conservation, so rare plants and endangered plants and sensitive species, and giving, getting data and giving information to land managers so that they can,
know what's on their property.
Is this rare species? Is it, are there other populations? Like, can we dig here? Can we not dig here? That kind of thing. Because there's a lot of oil and gas exploration in Colorado and fracking and stuff like that. And so they need to know, about the land. And so that's kind of one of the things we were doing at the Denver Botanic Gardens was applied science and using population genetics to answer some of those questions. So with my background, really the question is if I buy Blue Dream at this dispensary and Blue Dream at this dispensary, are they genetically identical or highly similar? And so that's where I started. And I approached my mentor for my master's degree at the population genetics lab and said, hey, I've got this great idea with cannabis. And he said, no, I'm not doing that. We're not doing that. But he went home and talked to his wife, and his wife is like, why not? Like, somebody's got to do it. Why don't you let her do it? And so he ended up talking to the dean of this college and the lawyers, and he emailed me and said, hey, will you come in and talk to me about something? And I didn't know. I thought it was another project that he wanted to talk to me about. And he's like, do you still want to do this project on cannabis? And I was like, yeah. And he's like, all right, you're in. So, they let me do it.
That's amazing.
So what was the very first project? What was it titled?
What was it? Weeding Out the Truth.
Weeding Out the Truth.
Oh, that's perfect.
I knew it's like a dissertation.
Yeah. So
I love it.
And then
they always make these really cool titles for like projects or dissertations and stuff.
Yeah.
So
that fits in.
Yeah, my first published paper on cannabis was Weeding Out the Truth, and there's a bit more to the title.
Right.
And I'm really surprised that my mentor let me do that, because he kind of, he's a professor, he's a professional, he's a scientist. They don't always have the best sense of humor, and puns usually are not part of, scientific papers.
Yeah.
But he let me.
Yeah.
And they have since published two more papers, and then I just have another one, which is what I have a poster downstairs on THC inflation.
That's right. You had the poster board.
And that paper all that data comes from has been accepted into PLOS One and should be published any day. Should have been published by now, but
what's
it's
PLOS One?
Oh, awesome.
It's PLOS One. It's a journal. It's an open access journal for scientific research. And so I'm super excited that's coming out. And the title of that one, he let me slide with Uncomfortably High.
I like that.
Yeah.
It sounds like a Pink Floyd song.
Like
the opposite of a Pink Floyd song.
A U2 song, maybe.
No, Uncomfortably Numb.
Oh,
so
like,
Uncomfortably High.
Uncomfortably High.
Pink.
Yeah, I think I like being comfortably numb better than
I like
uncomfortably
being
high.
Comfortably high.
I don't like being uncomfortably high. It's really uncomfortable.
I said comfortably.
Comfortably high.
Comfortably high, please.
So, wow. So you're like deep into the sciences stuff.
I'm deep in the weeds, yeah.
Yeah, so tell us, what have you found? What is like the surprising thing that most cannabis people don't know?
Takeaways. So the first study that I did, I went to dispense. So this was all from a consumer perspective too. Nobody knew that I was a scientist and I was going to be, messing in the lab with their cannabis. So I just, this was from a consumer perspective. So just like all of us go through with the experience, getting access to that same information. So what's available online, what's available on the shelf, what you get in the bottle is what you get. You don't have access to the plants to see how the plants are growing or anything like that. So I went around to different dispensaries and bought kind of just whatever I could find. I didn't really know that much about what was popular and stuff. So it kind of, after going to a few dispensaries, I kind of learned what most people were selling.
Yeah.
And so I got 30 different strains with multiple samples from different dispensaries to check to see if are all the Blue Dreams genetically similar, like we would expect. No, they're not. So of the 30 strains I tested, 27 of them had at least one genetic outlier. So, buyer beware.
And when you say genetic outlier, you mean like
it's not what like, okay, so picture a basket of Granny Smith apples, and they're all, they're all Granny Smith apples, and there's one red one in the basket. That's not a Granny Smith apple, right? It's something completely different. So that's the equivalent of finding a genetic outlier among these samples with all the same name.
So, but how did you know what was the actual blue dream to begin with?
I don't know what the actual blue dream is, and unless you would go directly to the breeder, you wouldn't have access to the original mother plant.
Right.
But when you have 9 samples and 8 of them are all genetically similar and one's different, you can kind of guess that maybe that one where it shows up 8 times is probably the one that is
gonna get close.
I guess the only person that will really get offended, well, it's not even person. The only thing that would take really great offense to that is the actual original blue dream plant, because maybe the wrong one's being replicated 9 times. And the original blue dream is like lonely and gone somewhere hidden away in a cave.
The original one is probably, I would imagine, I don't know, but like mothers don't generally last forever. I do know a few people who have had mothers for, up to 20 years.
And even
but
when,
that's not normally the case.
You take that mother and you make it into a smaller one, it's still not, it's just like a pheno 2 of it. It's not really like technically the same plant. Because you can grab if you're vegging a mother plant, you can take a little clone off of it and, continue its life. But at that same time, you're kind of creating a whole new. Right?
So it would still have this. It would be a genetic copy of the mother. However, there's other things that play into it. It's not just the DNA. There's also a thing called the epigenome, which can change. And plants when they undergo stress or when their environment changes, they have the ability to change their phenotype to adapt to a different environment.
Right. Okay.
So that's what they're doing. When you see those changes in clones, you're like, it didn't used to be like this. What happened? The genome didn't change. The plant changed its behavior.
Your environment.
How did Yoda end up
too.
over there? How did Yoda end up over? Did he fly over there?
He moved. He's got a mind of its own. It's Yoda.
I was like, what are you pointing at?
Sorry. No, Yoda was.
This is our mascot here.
Yoda has been paying attention. I put Yoda
He's
up here.
learning.
Yeah.
Well, you talking about endangered species, and Yoda loves that subject.
He's like,
He's like, I'm an endangered species. Sorry. So he was up here paying attention to you, and then he moved over there.
Yeah, he's got a mind of his own.
That
it's
threw
Yoda.
me off. That
That's
threw
okay.
me off. I apologize.
That's okay.
Okay, so sorry. Let's go back to the blue dream.
So people ask me, well, how do I know? Like, how am I going to know if I'm using Blue Dream as my medicine and I go to California or I go to a different dispensary than my where I use? Like, how am I going to know? The answer is you're not. You don't. You can't.
And they buy a seed offline that says Blue Dream. How do they truly know that?
They don't.
So how can we fix this problem?
So I mean, ultimately, and this isn't really a unique to cannabis problem. Before we had genetic tools to kind of look and identify things. Like this was rampant in the wine industry until we had genetic tools, because there were things that were, someone had the same thing as somebody else, but they had 2 different names. Or on the flip side, they had the two different things with the same name, or 2 of the same thing with different names. Like it was just kind of a mess, So
genetics is a mess. But it's
so
also
would it be right to
genetics
say
is a tool to help you figure out a puzzle.
it's the same.
Yeah.
Yeah.
Okay.
Would it be right to say, like, okay, you brought up
It's a
the wine
solution.
industry. So, there's different Chardonnays, different Pinot Grigios. Obviously, it's different because those companies add sugars to their product and stuff. But that plant that they're extracting from, the Chardonnay grape or whatnot,
it's a cutting. So it would be clone. So you would have, your 2 different grape cuttings. You have one in Italy and you take one to Napa Valley.
Would you say that's the same as to, the cannabis?
Yeah.
plant?
So there's an environmental factor that plays into a phenotype. And, as I said, plants have the ability to adapt to their environment. So if you put them in different environments, they're going to grow differently. They're going to express things differently so that they can survive in those different environments. So with cannabis, if I took a Blue Dream cutting home and you took one home, we would end up with completely different things and it could be, the skill of the grower. It could be nutrients, light, water, all of that stuff is going to play into the final product.
When tested, they would look like genetically different.
They would look genetically identical.
They would still be genetically identical. But you said they would be different. I mean,
Yes.
but not
So-
genetically.
Like smoke. Like I would say,
So
my opinion, like when you smell cannabis, some cannabises smell
Yeah.
different.
Yeah. So your phenotype, so what you look like and who you are physically
Yes.
is a product of both your genes, your genetic makeup, and the environment. Like twins don't look this like even like identical twins, they
Yep.
have the same genome. Never are they absolutely
That is
100%
the
identical.
best way to describe that.
You can have a twins, one's a smoker and loves to tan, and one is a health nut. You're going to see very, very different things because those things are a product of their environment. Same thing with cannabis, same thing with everything.
Mm-hmm.
So you kind of, like Daniela was saying yesterday at the keynote,
Mm-hmm.
the environment plays a significant factor in end product. So what are you getting? And we haven't really had the ability to assess to what extent does the environment play into the phenotype of cannabis.
And what about the nutrients?
Yeah, that's part of it. That's
yeah.
part of the environment. As well as harvest time. So if you harvest, something early versus late, you've got two different phenotypes, right? Because you've got the plant that's the flowers, the trichomes are maturing. You've got drying and curing. Storage conditions can also change the phenotype because terpenes are volatile. If you leave them out, if you do drying at the wrong temperature, you're going to lose some of those volatile terpenes, and so now you've got two different products. Time to sale, so how old is the product? And then how the
Shelf
customer
life.
treats it when they get it home. It's still changing over time.
So what do you do with all this knowledge and expertise? Because I'm thinking to myself,
I do podcasts and webcasts.
but but you
And
can really
consult,
help.
it sounds like.
I
You-
really don't.
Are you not
No.
helping any companies develop consistency and uniformity and teaching them the best practices?
No, not really, but I
Because
can.
it seems like that would be you'd be like a natural coach for like, because a lot of investors and businessmen that want to get into cannabis, but they need somebody, they need the expertise, and I'm listening to this, and I'm like, wow, this, I wanted to give you this. This is a MITA pin.
Oh,
'Cause
thanks.
You earned a MITA pin.
Aw.
Even
And
Yoda
initiated.
Yoda was happy that you were talking about endangered species, but your level of expertise is absolutely incredible. It's really cool to talk to you and just listen to all this.
So I do a lot of
Definitely.
education. So
Yeah.
I have also, in addition to all the things that I do, we've been developing education courses.
Right.
So I have one course called Flower Facts for Bud Tenders that kind of goes over everything very lightly. That would give a bud tender a really good background to talk confidently about the products that they're selling.
Right.
So
Which they should, yes.
yeah, and I mean, I have like a whole lot of. I think I have 38 different courses up on our website at the moment.
What's your website?
So you can find me and my company on my personal website, which is annaschwabe.com.
2 N's.
Yes, 2 N's. And I work for a company called 420 Organics. So if you go to their webpage, you can go to education and click on explore courses, and you can see all the courses that we offer there.
That's a web. I imagine that's a website that's worth exploring.
It is. I mean, I think it is, but I'm a little biased.
No, you're definitely on the right track, the right course. What's shore-organics.com?
So the company that I work for is 420 Organics, but we have two arms. So
Right.
we have a hemp arm, which is Shore Organics, and we have our marijuana side, which is 420 Organics. So because they're completely separate, but it's the same people who run both, because we want to keep our hemp and our marijuana separate.
That's amazing.
Kind of have to.
Are you doing the same sort of genetic? I mean, do you have like, the data, the information that you're collecting? What are you doing with all that? Is that all online or?
So I'm not actually currently actively collecting any data.
Yeah.
We will be. We have been in the experimental phases with it because we grow aquaponically, which is a little bit of a different system that almost all cannabis growers grow. In that it's a closed loop system. It's like an ecosystem. So we have fish.
I love that one.
We feed the fish, and then we have some biofilters that change the nitrogenous waste to a usable form for the plant. Then that water goes to the plants, feeds the plants. The plants pull all of the nutrients out of the water, and that clean water goes back to the fish. So it's completely closed. And we use very little water to do that, which is nice because the cannabis industry kind of uses a lot of water.
So it's like the fish shit you're using.
It is.
That's
What kind of
amazing.
fish do you use?
We have a mixture of tilapia, koi, and goldfish right
Koi
now.
fish. I love
Mhm.
that.
Yeah.
Is there any? I guess the science is still out as to what type of fish is the best, or is it just kind of mostly anecdotal stuff?
I don't have an answer. I'm
Yeah.
not the grower,
Mhm.
and my boss just kind of he's like the lord overlord of all the growing. And-
Overlord. That's a word I haven't
No,
heard in a
he's
while.
really nice. He's awesome. But I don't know nearly as much as I should about the whole aquaponics system. But also, I
Yeah.
know that we have proprietary setup, so it's probably
Mhm.
best that I don't know everything.
Is that over in New Jersey or?
Yeah,
We got to visit that sometime.
That'd be
we
sweet.
love visitors.
That would be
Yes.
amazing.
Yeah.
I've heard of, grows using fish it is what
Mhm.
they call it. And they actually have a company that
Yeah.
does that. But
I went to-
they have, it's literally a big bucket with the flour on top, and underneath you could see the little fishy swimming.
Yeah, there's a big one here in San Diego up on the highway.
Yeah, that's
I
cool.
visited it like 5 or 6
Oh.
years ago.
And a lot of the aquaponics that's around, it's a split loop. So they do have the fish in the water, and they do use that water to water the plants, but it's not all circulating in a closed loop.
Mhm.
That's what's
Okay.
interesting about our setup, because it's a lot easier to control. Like if we add anything, which we don't, if we needed to add more nitrogen or whatever, that would get circulated back to the fish, which can hurt the fish because they're pretty
Right.
sensitive. Or the bacteria culture that we have, we don't want to crash that because that throws everything out of whack. So we have to be really careful with our system. Whereas if you have a split loop, you can just take the water and water the plants. And then if there's something going on with the plants, you can treat the plants without worrying about, hurting the fish. So
Yeah.
that's a little easier to work with, but we don't want to do that because we like a challenge.
That's amazing.
I like it. You should call it the boomerang system because you're Australian
Right.
and it kind of comes around.
But I don't own the company, so.
I know. Well, one day.
So your background is more on the the lineage side of the Blue Dream
Yeah.
and the what is it, the genealogy of it.
Yeah. So population genetics is looking at relationships between individuals, among individuals, among groups of individuals, among populations, among species, closely related species, things like that. So, for example, we can look at populations of cannabis that are feral here in the United States versus
Right.
in Afghanistan or Russia or whatever. And there's a few papers out there if people are interested in population genetics, looking at like the evolutionary history and how it moved around the globe, which was largely done by people moving it around the globe.
Are you familiar with the website, the Seed Finder?
Yeah.
EN Seed Finder.
Yeah.
So that's kind of where I base. Like I'm looking at Blue Dream right now, and it says Blue Dream started. Actually, go at the bottom. It's Hawaiian. It's a sativa,
Yeah.
and then it moves to now it's a purist indica. So that when it says purist indica times
So-
Hawaiian,
Let me check this out. So,
She's got a
and that's
correct it.
another thing that I want to move into too. That this big concept in the cannabis industry right now is indica versus sativa. And then there's the hybrid in the middle.
This is how it was developed. So it started. This is like going back to like great grandparents.
Very, very
and then
first.
as you move through the family, it was crossed with a skunk, and then with a haze, and then it was somewhere back in the history there's
This here,
Afghani.
so it says Afghani. Does that mean it was somebody found a breeder seed from
I don't-
Afghani?
I find this really hard to read, but there might have been a strain that had all of these lineages in there, and then they crossed this with a blueberry, and then Blue Dream was born.
Mm-hmm.
So, I talk about cannabis strains like dogs because everybody's familiar with dogs.
Yeah.
Everybody knows what a mutt is.
The breeds.
It's a mixture of all different kinds of
All
dogs.
a whole bunch of yeah.
Right. So that's what a lot of cannabis.
She's a mutt.
I think
Apparently
most-
today
I think
I'm
most
looking
people
like a
are.
mutt.
I think
I'm
most
a
people
mutt.
are mixtures of multiple different kinds of people from different places in the world, right?
Yes.
23andMe will tell you like you're 20% Irish and 30% Mexican and whatever.
Yeah.
So that's population genetics. So with dogs, like you got two, you go and you adopt two mutts. They're both beautiful dogs, but you can't really tell what they are.
Yeah.
And you love them so much and they're so cute. You decide to breed them. The puppies are all going to be different. They're
Yeah, I got
not
a
they're not going to look
bulldog
like each
and a-
other probably, and they're probably not going to look like their parents.
Bulldog
because-
and a
Or
boxer.
mutts.
Yeah. So you're going to have this mishmash of whatever the parents had in them. And then, you pick your favorite puppy, and that's the one you choose to keep, and you give all the other ones away. And you call this one that you love, you call him Charlie, right?
Mm-hmm.
Charlie's not a breed. Charlie is a dog. But he's a
right.
thing. He's a thing, right? He's got a name, and you love him. You can't go to the AKC and say, I want to register Charlie as a new breed.
Right.
You have to do more work. You have to make more Charlies. And to do that, you have to breed Charlie, unfortunately, probably with one of his parents.
Yeah.
And then his mother would be probably the likely candidate. And then you might be able to get some more Charlies in the next litter, and you would pull those out. And then you would start crossing those and weeding out the phenotypes that are not Charlie. And then at some point, you're going to get to a point where you put two Charlies together and all they have is a litter of Charlies. Then you can go to the AKC and say, I have developed a new breed. That's the equivalent of a cultivar, right?
I want to be my own breed.
You have a lot of work to do.
Yeah, I know. I'm just trying to figure out the steps that I need to do to get there.
But
I think you could help me.
With people and even with dogs, like there's a lot of ethical issues in doing that. Like inbreeding, not a great idea with,
How could I inbreed
mammals.
myself somehow? You see what I'm saying?
Probably.
So if I believe that I'm like genetically perfect, and
I mean,
want to make sure that all
you
future
could take
human beings are
you
like
could take
me.
2 of your sperm and go to a lab and say, put these 2 together and make another one
Yeah.
of me. Or you could clone yourself.
I think I have to clone myself,
You
right?
could clone
not
yourself,
really
and then you
breeding.
could breed your clones together.
Yeah,
Yeah.
make a clone in the clone.
Yeah, because I wouldn't
in a
want
clone.
to. I wouldn't want my genetics to be susceptible to the imperfections of a woman. So I mean, not just
This guy.
not just. I mean, I'm nothing against women. I'm just saying,
You just want
my
to make a man beast?
genetic.
You don't want to dirty it up with
I'll
all these
just-
X chromosomes.
I know.
I'm making a joke,
God.
but if you think about the parallels and how we do this with animals and plants and humans, there's a lot to the philosophy of this concept.
Yeah.
So
back to cannabis, like a lot of
Although
cannabis
maybe it
strains
might
that
be
we have are not stable.
genetically
And that's
perfect.
why we clone, because that's really the only way that you're going to get an exact replica of
Right.
that plant, because the genetics aren't stable.
exact replica
Even
of
when
me.
you have 2 blue dreams and you breed them together, there's still a lot of diversity in those lines, and when you put those 2 together, you're going to get a whole bunch of stuff that you weren't expecting.
Maybe this is why I haven't had kids yet.
Is this why?
He's
'Cause
having
I'm
like
worried
a
about
life
allowing
realization over here.
there to be
Getting kids you don't want, some
No, because anything
a short
beyond
one
me
and a
would be a genetic imperfection.
well, then you're at the end of the line of perfect.
Right. I am
Best
the
to
end
stop there.
of the line.
Get lots of pictures.
This all
'cause
makes
the
sense
line
to
ends
me.
with you.
Like
This has been one of my
from Anna
favorite
herself.
We're going to hang out more because I'm learning a lot from you, but we do got to get it wrapped up.
Yep.
We want to do a full hour with you sometime. We're trying to keep
Yeah.
the 20, 30 minute here.
That was just my first experiment. I've got 4 more.
Well,
and I'm sure you have more on the way. Many projects
Possibly.
that you're
uh,
working on.
Epigenetic project,
Oh
doing
my
clones
gosh.
of clones of clones to see if the epigenome changes through multiple cloning iterations.
Right.
That has not hit. That's being. Hopefully, we're getting that analyzed soon. It's been sitting on the shelf for a while.
And what is that? What is that word?
The epigenome?
Yes.
So epigenome, epi meaning on top of, genome, so it's on top of the genome. So the genome stays the way that the genome is because you can't really mess with, not to get too technical, but
Okay.
it's changes on the surface of the DNA that lead to changes in expression. So you can basically stick, like think of a thread. That would be the DNA. If you tied beads on that thread, now there's pieces of DNA that are going to be easily accessible to express and things that are inside the bead that you're not going to be able to access. And that can lead to heritable changes. So even though you didn't change the DNA code,
Right.
those heritable changes can be passed on to the next generation. And it's pretty common in plants because when plants encounter environmental stresses, they can't just get up and leave, right?
Yeah.
And they want their offspring to survive, so they have a way to change their epigenome so that their offspring would then have a better chance of survival if they make it to reproductive age. Their offspring would have a better chance of survival because potentially that environment is still not great, and then the offspring have a better chance of survival. And those epigenetic changes can go away as well, or they can lead to permanent changes.
You know what's frightening to
The
me?
epigenome is crazy.
You know what's really frightening? Is that her knowledge, as she puts it more onto the line, and it becomes more and more digital, then it belongs to AI. And once AI takes her knowledge and then moves in a direction that it wants to, rather than in a direction that we want it to, that's like the beginning of the Matrix.
And that's what we need, though.
Well,
Because
I mean, maybe. I'm
AI
sorry,
is
but
the future, and we need this,
you know,
kind of information
every time
to
I
get
realize,
out there.
every time I talk to the scientists and the doctors and the PhDs, and this was what I realized yesterday with the other guys, is that, you know, they're creating all this knowledge, they're breaking it down, they're helping us understand everything, and they're putting it on the line, and then eventually AI will have access to that, and AI is going to use that to take over us. So you guys are inadvertently, because you're so smart and so intelligent.
It sounds like we know a lot, but any scientist that you talk to is going to tell you that the more we learn and the more we know, the more we realize we don't know
No,
anything.
agreed. It's okay. It's okay.
No,
It's inevitable.
So
I'm not putting like a judgment
on
on it.
keep
Yeah.
learning, and
Yeah.
so if AI picks it up, that's fine because
Yeah.
it's still
That's
just
what I'm
scratching
saying,
the surface of
Yeah.
everything we don't know.
Literally,
think-
we are what? How many years are you into your research?
So I started cannabis specific research.
Yeah, cannabis specific.
I started in 2015, so 8 years, and I don't know anything.
And see,
No,
you're
that's
somebody
okay,
that knows
sorry.
more than all of us here combined. So you saying that means there's just so much out there.
Well, and it's so interesting, and I know this is true in any industry that people sound like they know so much, or they are very confident that they know a lot.
You know a lot more than I do.
All of us. Put
There's
together.
a lot of
That's for
overly
sure.
confident people.
She's like, a lot of people
think they know
you.
a lot, but really they don't know. And it's the people who don't know that they, that think they know, and it's like you have no idea. But anyway.
Yeah.
No, this is great. We're learning a lot. The people out there that watch us can learn a lot from you. And if they want to dive in deeper, they can call you. They won't call me.
Oh, we'll
They'll
do a
call
deeper dive here with her.
Yeah, but they'll call you directly.
I mean, if anyone is interested in all the stuff that I didn't talk about today, or even the stuff that I did talk about, it's all available on my website. Like my dissertation from when I was a baby scientist is published, and you can read the 200-page stuff. But it also my research papers that have been through the peer review process, you can read those. And I try to keep it really, like, I know scientific papers can be really difficult to read with jargon and stuff like that, but I try to keep it so that normal people can read it without having to look up every second word, because that's just annoying.
Oh, that's amazing.
Well, Anna, it has
Dumb
been
down for us.
insightful, and we appreciate you being here. And we have to keep it under 30 minutes. And so, Anna Schwabe, we will have her information on how to get a hold of you. We thank you for joining us.
Thanks for having me. This was really fun.
Yes, no,
I'm
you.
a huge fan.
I feel like my mind has expanded just
I
listening
can
to
talk
you.
about
So,
cannabis for days, like literally.
love you guys. Thank you very much.
Thank you so
Thank
much.
you so much. And that was another episode of MITA Unshackled.